Nobody told me this before I started working in a lab, and honestly, it would have saved me so much time and stress and many mistakes if someone just sat me down and had been real about what lab work actually looks like. So, if you're about to start a placement, a rotation, or a PhD, watch this first. Hi, if you're new here, my name is Aku and I'm a second year Neuroscience PhD student studying in London. And I've been working in research labs for quite a few years now, and I've learned that there's a massive gap between what you learn in your undergrad labs and what actually happens in a real research lab. And that gap is exactly what this video is about. So let's get into it. The first thing, lab work is actually a lot of waiting. Like, a lot. You're waiting for an incubation, you're waiting for the centrifuge to finish, waiting for your cells to be confluent enough. It's generally one of the most subjective measurements in biology. I remember one specific afternoon where I had an experiment and it was meant to take me about an hour, but 1 hour turned into 2 because the water bath took forever to heat up, then 2 hours turned into 3 because I realised I was missing a reagent and had to go find it, and just like that, half my day was gone. And I only did one step. Honestly, this is not a bad thing once you learn how to work with it. The best researchers I know are masters at filling their waiting time. They're not doom scrolling on their phone, they're writing, reading papers, or planning the next experiment. So, the move is always have a secondary task running. Never just wait. Use it, because lab time is expensive and your time is expensive. The waiting is part of the process, not a break from it. This brings me to the second thing, and this one sounds obvious, but I cannot stress this enough. Write everything down. And I'm not just talking about the steps of your protocol, I mean the date, the time you started, the lot number of your reagent, the passage number of your cells, the exact volume you pipetted, how the cells looked under the microscope before you even touched them. Anything, everything. Because here is the thing. 3 or 6 months from now, when your results look strange and your supervisor asks you, what was the passage number of those cells? You either have the answer or you don't. And those are two very different conversations to be in. In one conversation, you might say, passage 14, and then it moves on quickly. In the other version, you say, I don't know, and now your experiment is not reproducible. And reproducibility is the entire foundation of science. If someone cannot repeat what you did and get the same result, your results effectively don't really exist. And that's how serious a lab notebook is. Third, this is not always true, but it did genuinely mess with my head when I first realised it, and that is, protocols can sometimes just be a starting point. Not a guarantee. You gotta find a protocol and you gotta follow it exactly. Every step, every volume, every timing. And sometimes it's just not gonna work. And this is one of the most disorientating feelings in early lab life. Because you did everything right. You followed the instructions, but it just didn't work. But that's the thing about research. That is kind of the point. You're working on the edge of what's known. If the protocol worked every time for everyone, we would already know the answer, so you technically not even need to be here. So, when something doesn't work, and it will not work, your job is not to panic. Your job is to troubleshoot. Change one variable at a time. 1, not 5. If you change 5 things and it works, you have no idea which one fixed it. That's not science, that's luck. So, ask your lab mates, ask your supervisor, and look at the literature. And honestly, some of the best learning, I mean the learning that actually sticks, happens when something goes wrong and you have to figure out why. So reframe it. A failed experiment is not a failed you, it's just data that didn't work. And that's still data. Fourth, and that is learn when to ask for help. This one did take me a bit long. There's a sweet spot between asking for help every 5 minutes and struggling alone for weeks. But it's important to remember, asking for help is not a weakness, it's actually efficiency. The postdocs who have been working in your lab for years know way more than you could. That's not a gap in your ability, that's literally just time. So, I made a rule for myself. The 30-minute rule. If I've been stuck on something for more than 30 minutes and I'm not making any progress, I go ask someone. I don't let an entire afternoon disappear into a problem someone could have solved in just 5 minutes. And honestly, most experienced researchers actually like being asked. It makes them feel like the expert they are. And remember to write what they say down. And finally, small wins generally make your day. And I'm not talking about publishing a paper. And I'm not talking about getting results what will change your field. I'm talking about your gel ran clean, your cells look good, your PCR worked on the first time, you found the reagent you needed in the freezer within 30 seconds. In undergrad, you're graded on big outputs, exams, essays, final projects, but in the lab, most days do not have this. Most days are just simply pipetting, incubating, waiting. And if you only celebrate the big things, you would spend most of your time feeling like nothing happened. So, let yourself feel good when something small goes right. A clean gel is a win. Cells at the right confluency is a win. Because research is hard enough already, and you don't need to make it harder by refusing to acknowledge when something actually worked. The small wins are what keeps you going between the big ones. And here's a bonus one, but I do think is important, and that is, you probably will at some point feel imposter syndrome. If you feel like everyone around you already knows more than you do, if you feel like you've somehow tricked your way into the position and any day someone's going to figure out, that's not a sign that you don't belong. That's a sign that you're new. That's it. Every person in the lab had their first day, and every person had a moment where they had no idea what they were doing. Everyone looked at protocol for the first time and thought, what does this actually mean? Feeling imposter syndrome is not a red flag. It's a sign that you're actually learning something real. If you walk into a lab and everything already makes sense, you're not even learning. You're just confirming what you already know. Discomfort is part of the price of growth. So, feel the discomfort and keep going anyway. So, these are the things I wish someone told me before I started working in a lab. If this helped you, or if any part resonated with you, please give it a like. And if you're new here, I post about PhD life, neuroscience, and academic research. Subscribe so you don't miss the next one. I'll see you next time.